So let's move back to the back of the eye. Just to remind you that we have six program areas at the NEI. The SAVP program is the second largest. Within SAVP, there are seven themes. I'm going to tell you about two of these themes. The oculomotor systems, which essentially deals with the biology of eye movement, and then strabismus and amblyopia, which essentially deals with the pathobiology. We start with the oculomotor systems, but before that, let's get some background. Eye movement can be classified in many different ways. Perhaps the easiest one is to think of it as fast and slow. Fast eye movement, also called saccades, essentially refers back to the work of classic work of Jarvis. I'm sure you've seen a figure like this. And the idea is that if you track eye position, you'll find that the eyes are constantly moving their fixation point, usually to the most salient point, such as the eyes. This is constantly happening. In fact, it's about 3 times per second, upwards of 183,000 times a day. Now, these saccade movements are what's called ballistic. They're very, very fast, about 500 to 600 degrees per second. Of course, it doesn't go for an entire second. It goes only for about 12 degrees per saccade, but they're very, very fast. Once they start, they cannot stop, even if you change the target. On the other hand, you have slow eye movement, and two main kinds, smooth pursuit. This is when you have a target that's moving, and you want to keep track of that moving target. Then you follow on a different type of eye movement, a slow eye movement. It's much slower. It's about 10 times, more than 10 times slower than the saccades. Of course, the key for all type movements, fast and slow, is to keep the target on the most sensitive part of the retina, the fovea, which is only a tiny, tiny part, only about 2 degrees. Okay, so who's doing all these movements? By the way, I'll talk about versions in a bit. Who's doing these types of movements? Muscles. There are 3 pairs of what's called extraocular muscles. These 3 pairs you find if you want to do the vertical eye movement. So you'll have what's called the superior rectus and the inferior rectus. Well, if you want to do your eye wants to move sideways, horizontal, then you have the medial rectus and the lateral rectus. The eye is also able to do a sort of a torsional movement. That's done by the superior oblique and the inferior oblique, 3 pairs. And who's controlling them? 3 innervating nerves. The main one is the oculomotor nerve, the 3rd cranial nerve. Except for the superior oblique that is driven by the trochlear nerve, the 4th cranial nerve, and the lateral rectus that is driven by, I had some arrows to point, this is the superior oblique, and the lateral rectus that is driven by the abducens. Actually, their names, that's what they mean. The abducens just moves the eye away from the midline. Now, there's been quite a few success stories of NEI vision, of NEI-funded research in this area relating to ocular motility. I'd like to highlight the work of Dr. Elizabeth Engel and her colleagues. And so she's interested in this particular syndrome, CFEOM3, congenital fibrosis of the extraocular muscles type 3. And so they identify these families. Some would have some subjects with this particular mutation. And when they examined with MRI, they found that the oculomotor nerves were hypoplastic. They are not fully developed. And so when they mapped the mutations, they found that it actually affects the tubulin beta gene. And this tubulin is neuron specific. They identify a number of mutations and that affect a number of different amino acids, including some in the, at least among the newer identified mutations in the kinesin binding site. And so they wanted to model this. And so they picked one particular mutation, the R262C mutation. They modeled it in mice. And through some really intricate analysis, very detailed analysis, we're able to find what's going on in these with this particular mutation. It turns out that the superior part of the oculomotor nerve doesn't quite get there. It is the hypoplastic, it's not fully developed, which means the innervation of the superior rectus is affected. Moreover, it also affects the muscle that is controlling the upper eyelid movement. that's the same branch. And sure enough, these patients have eye drooping. So it's a nice, complete story of going at the molecular level. What about the muscles? More success stories related to the muscles themselves. This is from some beautiful work by Dr. Joe Deemer at UCLA, and in which he came up with this idea of compartmentalization of these extraocular muscles. The idea being that different domains of the muscles are being innervated separately. And so he demonstrated it in a number of muscles. In this panel, you see it for the human lateral rectus muscle that's, of course, innervated by the 6th cranial nerve. And as you can see, the inferior division, highlighted in green here, makes a posterior entry. And yet the superior division in magenta here makes a more anterior entry. And so the cool part is that there is no overlap in the innervation. Dr. Deemer also demonstrated this for the in the bovine model for the superior oblique muscle that's innervated by the 4th cranial nerve. And as you can see, and he's done some beautiful 3D reconstruction. And you can see that the inferior division of this nerve versus the superior division, and there is no overlap in these territories. The key part of this finding, obviously, is that it gives the extraocular muscles a more detailed repertoire, more detailed control of these muscles. Okay, let's move on to the eye movements. The main eye movement, obviously, is the saccade. And so a key new development was the introduction of the optogenetics technique to try to control or manipulate eye movement. Just very briefly, what optogenetics is essentially to take this protein, channel rhodopsin, belongs to the same family as rhodopsin, except in itself is an ion channel. It's light sensitive like rhodopsin, except in the channel rhodopsin itself is a light channel. And so when you activate it with a light, in this case it's sensitive to the blue light, you can actually generate a neuronal response. And so this group at 3 funded NEI investigators at the University of Washington developed this technique for the non-human primate, and were able to inject this particular channel rhodopsin 2 using the AAV vector, right? And using this particular promoter just to affect the Purkinje cells. That the MTRA is just to keep track of where your construct goes. And when they did that, tested these monkeys on the saccade tasks, they found that they can perturb, which is most clearly seen in the vertical direction. And you can see that circles, these are 2 circles between the control and the injected monkey. So this is really cool tool of being able to finally dissect these eye movement pathways. Let's move on to slow eye movement. We mentioned smooth pursuit. Now, smooth pursuit has been fairly well studied to the point that actually Dr. Lisberger, in NEI-funded work, has used it to study more generally the idea of motor learning. And so he came up with this paradigm, really cool paradigm. And so essentially he has this monkey, or number of monkeys, but looking through pursuing a particular stimulus in the horizontal way, and then abruptly change the direction at it, right? And so what happens, obviously, if you look in the bottom figure, you'll see that at first the monkey kind of misses the target. And you can see its eye movement is different from where the target is. By the second trial, the monkey kind of anticipates something going on. So it's a little bit better, but still, it still misses it. Now, by the 100th trial, the monkey is figuring out what's going on here with this trial, what it's supposed to do. Still not perfect. Dr. Lisberger does this analysis for these trials for over 1,100 trials. And by the 1,000th trial, look at how accurately this monkey is able to predict where the motion of the stimulus is. And so this is a summary of the different trials. What this allowed Dr. Lisberger to do is to identify two different components of pursuit learning. In the first component, which rises rapidly, it takes you about maybe the first 100 trials, that's in the green trace, you can get most of your learning ability. But still, if you want to get really high up there to the blue level, then you would need go up to the 1,000 trial to get the accuracy up. Okay, another type of slow eye movement is vergence. Now this is a disconjugate, unlike this pursuit and the saccades where the two eyes go in the same direction. This is where the eyes go in opposite directions. So if I'm looking at you and then I decide to focus on the screen, my eyes actually are going to converge for the near target. If I decide to go look back at you, then my eyes will diverge for the far target. And so we have some really cool studies recently published. This is work by Dr. Mark Walton. What he did was to screen these monkey populations and identify the particular monkey that is naturally affected or has vergence deficit. You can see in the left figure for a normal monkey that's asked to do this vergence task, both diverging and converging. It does very well. Not exact, but does very well. Compare that with this mutant monkey. It's very, very deficient in both tasks. But here's the cool part. When he gives the same affected monkey a background that a little bit helps with the 3D, with this checkerboard pattern, the monkey, this affected monkey, is actually able to do more or less like the wild type. Now, the beauty of this system is obviously, now that you have a non-human primate, is that you can do a lot of electrophysiological studies. Okay, so that was for the eye movement part of the portfolio. What about the disease part? Right? And the main disease obviously affects strabismus. And in strabismus, it has about 3% prevalence of US children show eye misalignment. There are many different ways of classifying strabismus. I'm going to follow Horton's approach, which is 3 main types. There is what's called the infantile esotropia, which is, as you can see, the eye is turning inward. Now, this develops, it's convergent. It develops by 6 months, and this one doesn't have very good prognosis, not very good ability to treat this. On the other hand, you have another type that's called accommodative esotropia. Again, the eye is moving inward, except when you simply correct this large refractive error, the eyes pretty much look normal. This is very treatable. There's a third main type. It's called alternating exotropia. This is going, obviously, this is divergent, and it's intermittent. So, and it's when you see a video of this, it's really striking how the eye movement happens. Now, the thinking in the field is that it's the balance between these muscle factors and these brain factors that drives this strabismus phenotype. And in NEI-funded work, this is by Dr. Vallabh Das, he has this monkey where they actually did this surgical treatment. They, first of all, they grow these mature these monkeys with exotropia by having them wear prisms. And once you have that, then you go back and do the surgical procedure that you would do normally on a human subject, which is essentially you want to weaken, you want to strengthen the weak medial rectus muscle by resecting it so that it's shorter, and you want to weaken the strong lateral rectus muscle by kind of moving its point of insertion so it cannot pull as much, right? And so they did that and got good results in that. They were able to get 70% reduction in the strabismus angle when they did that. Except here's the bad news. When within 6 months, these monkeys, they did it in 2 different monkeys, these monkeys essentially reverted to the same strabismus that they had before. And worse yet, when they tested the neuronal drive, the medial rectus that you wanted to strengthen, well, its neuronal drive actually was reduced. Moreover, the lateral rectus, which you wanted to weaken, its neuronal drive increased. So plasticity is a double-edged sword. Amblyopia, the different types of amblyopia. In terms of treatment, these are essentially you want to try to fix the refractive error, or you do the occlusion, block this dominant eye, or you want to do again penalize the dominant eye by adding some atropine, or vision therapy that I'll talk about in a bit. Now, we have a number of success stories in this program, actually including one by Dr. Alonso, and which showed that, very recently published, that the on visual pathway is more effective than the off. This is the light versus dark, and so this is a potential to where you'd want to focus your therapy. What I would like to move on to is this central theme in amblyopia, really in the field for the past 60 years, which is the idea of critical periods, which is there's a particular interval where the nervous system is particularly sensitive to stimuli, right? And if you miss that particular window, then you cannot develop the function anymore. And so a newer idea now is the idea of maybe there are some breaks to define these, and if there's a way to try to remove these breaks. And with that idea, some work by some NEI-funded work by Dr. Mark Baer showed that if you just simply temporarily disable the retinas so that the animal doesn't see anything for some time, and then reactivate it, you'll see that actually you can open up this plasticity period. This work is actually built on previous work by Dr. Betsy Quinlan at University of Maryland that showed that simply by rearing these animals, put them for 7 days, 10 days in total darkness, and then light reintroduction, it opens up this plasticity period. And so she provides this nice model for the different ways you can approach to try to open up the plasticity. I'm going to, in the interest of time, just move on to essentially towards the last, which is the point of neuromodulation. If you see under neuromodulation, it talks about video game training. Well, that gets us to the point of stereopsis. Now, obviously, which is 3D vision, which is essentially the main benefit of having two eyes, being able to see in three dimensions. And so using this, our council member, Dr. Dennis Levi, is actually working on this area, and he's able to use these off-the-shelf video games, 3D video games, in adult amblyopic subjects. Some of them have strabismus, some without, and able to get some really very impressive improvement in the visual acuity for these subjects. All they need to do first to kind of weaken the strong dominant eye, have these subjects practice this video game for about 4 to 6 weeks, maybe a couple of hours per session, and get some very good response. Dr. Levi is also obviously interested in children. He's got some very positive results in that domain as well. He's interested now to try to come up with this custom-made system for children to get hopefully even higher response rate. And so, as you can see, we have a number of advances in the field. I talked about some of these. There are some more success stories, but the opportunities are there, and obviously the challenges still remain. And so I hope you found this overview helpful. And in terms of the rest of the SAVP program, we'll hear from my colleague, Sherry. Please.